Welcome to OA Week 2024. We're going to get started in just a few moments. Okay, awesome. We're going to go ahead and get started. Thank you everyone for joining us here at OA Week 2024. My name is Elise Keister, and I'm one of the GOA-ON secretariats. Please note that this session is being recorded. OA Week is presented by four sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA OAP, the United States National Oceanic and Atmospheric Administration Ocean Acidification Program. Third, the IAEA OAICC, the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And last but not least, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. GOA-ON was established in 2012 with just a handful of members. Since then, GOA-ON has grown immensely. It now has over 1,200 members from 114 countries. GOA-ON also consists of several regional hubs, which span across continents and oceanographic regions. We'll be hearing from most of them throughout OE Week. If you are not a member yet, I encourage you to go ahead and go to goon.org and consider becoming a member. So OE Week debuted in 2020 and then returned in 2021 when events and conferences were still being postponed due to the COVID-19 pandemic. Following the successful in-person Ocean in the High CO2 World in 2022 and another successful OA Week 2023, GOA-ON is bringing back OA Week 2024 to maintain the momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of ocean acidification topics and speakers from around the world. During the presentation, all participants are in listen-only mode. You are welcome to type any of your questions in the question box, which can be found at the bottom of the control panel. We will be monitoring incoming questions and we'll pose them to our speakers during the Q&A session and after all the speakers have spoken. And during that Q&A discussion at the very end, you also have the option to raise your hand and ask your question directly. And with that, I would like to introduce you to our two moderators for this session, which are Martin Hernandez Ayon. So he is an investigator at the University of Baja California Instituto de Investigación of Oceanography of the Autonomous. And he's also the co-chair of the North American Hub Steering Committee. And then also Anna Hughes, who is a PhD student at Oregon State University in the Department of Earth, Ocean, and Atmospheric Sciences. And she's also the ICONIC, which stands for the International Carbon Network for Early Careers, representative of the North American Hub Steering Committee. And with that, Anna and Martin, the floor is yours.
Excellent. We want to say thank you for the introduction. And we also want to say thank you to all the audience for tuning in around the globe for this section is North American Hub session. We have three excellent speakers, one from US, one from Canada, and one from Mexico. But also we have the welcome from Dr. and friends, Dr. Richard Feugere, and I'm going to request to Anna, please introduce our Dr. Feugere, please.
Yes, Dr. Feugere will give an introduction to the presentation. So it is my pleasure to introduce Dr. Richard Feugere. Dr. Richard Feugere is a senior scientist with NOAA's Pacific Marine Environmental Laboratory in Seattle. His major research areas are carbon cycling in the oceans and ocean acidification processes. He received a BA in chemistry from the University of St. Thomas in 1969, and then went to Texas A&M University, where he received both a MS degree in 1971 and a PhD degree in 1974 in chemical oceanography. So thank you, Dr. Feugere.
Well, thank you very much, Martine and Anna. I really appreciate that introduction. I wanted to introduce myself. I am one of the four co-chairs of the North American Hub. The other chair members are Helen Gurney-Smith and Brent Els from Canada, Martine Hernandez Ayon, and myself from the USA. So we're very been working together with our many colleagues, as much as 120 members. And for the last 5 or 6 years. And so it's been a well-developed regional hub doing all sorts of different kinds of collaborations, both in the laboratory, in the field, and with synthesis efforts. The next part, at least. Yeah, so as I said, we're 120 members, and about 30 to 50 members attend our biannual meetings. And from about 2023 on, we generally have about 2 hub meetings that are virtual and 1 in-person meeting that's every other year. And so we've been maintaining that. We also have a data portal effort that is maintained by the University of Washington, GOA-ON data portal. And we also contribute to the OR's contributions in development of synthesis products. Next point. So our hub is working strongly together to provide synthesis products and synthesis presentations. And we'll hear a little bit about that from John a little later. And we plan for the development of the Marine CDR continuation for the international group. And we have face-to-face meetings in Canada, Mexico, and the United States. Next slide. So we have 8 major priorities that we have for the North American Hub. The first one is to interact and coordinate on cruise plans. We've been fortunate enough to work with our colleagues from Canada and Mexico to have cooperation and interactions on the cruise. Many of our cruises involve colleagues from our member nations, and so that's worked out really well, and we do our planning at our annual meetings. We also create a comprehensive inventory and data exchange through the North American Hub using the GOA-ON data portal. And that's been very, very successful at accumulating all the data that we have around North America. We also have been very much involved with the OR's outcomes, and the North American Hub members have been participating in 3 or 4 major components of the outcomes. We've also been extremely involved in developing and the coordination and the development of synthesis products for the North American Hub region in particular, but also for the global regions as well. And so these synthesis products provide us with the kinds of large-scale picture of the acidification trends and processes. And we've been able to develop the climatology, and more recently been working to develop new indicators that are on a monthly basis. And John Sharp will talk about a little bit about that very shortly. We also are developing new synthesis technology for the GOA-ON community, including time series analysis, algorithm development, and other approaches to, again, provide new synthesis products that weren't available until we came together on this. The North American Hub has been a leader in developing and maintaining the CRMs for the global community under the auspices of Dr. Andrew Dixon from Scripps. And we are extending that effort into the future with collaborations with NIST and other colleagues from Scripps as well, and also extending the capabilities of the CRMs to address issues in estuarine and coastal waters. We have been encouraging collaborative laboratory and field studies with other hubs, and as well as through the peer-to-peer process, and which has been very successful. And we are encouraging all of our communities and all of our hubs to interact even more in the future and develop a lot more interactive efforts than we have had before. And our last effort will be planning the next virtual meeting and face-to-face meeting at St. John's in Newfoundland in Canada in June. So we have lots of activities going on. It's a very exciting time for us to be interacting with our newest hubs, and we're really excited to do so. Next slide. Just an example of some of the new synthesis products we've been able to develop is the recent work by Liqing Zhang and to develop what we call the CODAP North America, which is a development of climatologies for North America of ocean acidification parameters, such as aragonite saturation state and hydrogen ion concentration, FCO2, and other carbonate species determinations, and looking at this from a long-term climatological mean perspective at many depths. Now, we're hoping to expand this effort in the future, and as I said, Johnson Sharp will show us how we can develop new technology to just that. Thank you very much. I will now turn it back to Martine and Anna to introduce the speakers.
Excellent. Thank you, Dr. Feugere, for this introduction. And we're going to start with the USA, with the participation of Jonathan Sharp. He worked for the NOAA, for the Pacific Marine Environmental Laboratory. John is a chemical oceanographer with a special interest in carbon, oxygen, dissolved nutrient cycle through the global ocean. And he actually is involved with the observational data from ship-based surveys and autonomous platform to study spatiotemporal variability in ocean acidification and biochemistry. Thank you, John, for your participation, and the floor is yours.
Awesome. Thank you very much. Let's see here. Do we have a full screen presentation?
Yes, it looks great.
Okay, great. Thank you, Elise. So yeah, thanks very much, Martine, for that introduction. My name is John Sharp. I'm a research scientist at the Cooperative Institute for Climate, Ocean, and Ecosystem Studies at the University of Washington. But I sit here at NOAA Pacific Marine Environmental Lab. And so I'm going to speak today about some ongoing work that I'm leading that attempts to quantify regional indicators of ocean acidification by mapping carbonate chemistry observations at regional scales. But first, I'll speak a little bit more generally about observational quantifications of ocean acidification at the global scale, and some work that my NOAA colleagues and colleagues at PMEL are doing. So in prepping this talk, I went to this issue of Oceanography from last year that took a look at the science from NOAA's Pacific Marine Environmental Lab over the past 50 years, just to pull some examples of how NOAA scientists and PMEL scientists in particular contribute to observations of ocean acidification. So at the global scale, our scientists participate in global hydrographic cruises where carbonate chemistry parameters like dissolved inorganic carbon, total alkalinity, and pH are measured. We do regional studies looking at vulnerability of species to changing ocean conditions and ocean acidification. This is an example from Simone Allen's work in that issue last year, looking at Dungeness crabs off the Olympic Peninsula. And PMEL scientists are leaders in autonomous technologies and looking at emerging trends in ocean acidification. So this is from Adrian Sutton's work looking at the global network of moored buoys with CO2 sensors. And this is a figure from Dr. Feely's paper in that issue, which gives an idea of the spatial coverage of ocean acidification observations at a global scale and across a few different time periods. And this looks at both autonomous FCO2 measurements, CO2 fugacity, and discrete dissolved inorganic carbon, total alkalinity, and pH measurements. So this article stated that we now have more than 34 million carbon observations to draw from to establish a record of decadal changes in the surface ocean carbonate chemistry system, which is really a fantastic observational record and is due to very hard work from so many different people over so many years. And that record is helped tremendously by the efforts of those who maintain the Surface Ocean CO2 Atlas, which is a quality controlled synthesis of surface ocean CO2 fugacity measurements. These two figures are from the most recent release poster of SOCAT, which looks at the spatial distribution of observations between 1970 and 2023, as well as the annual number of observations color-coded by the SOCAT release year. And they actually made a point in this poster that we're seeing a bit of a decline in the amount of surface ocean carbonate chemistry observations over the last 5 or 6 years, which I think is an important thing to keep an eye on and highlights the need to sustain funding for these types of observations. And so we use SOCAT for the work that I'll be describing today. The SOCAT data synthesis effort also allows for the creation of these kinds of synthesis mapped data products that Dr. Feely was mentioning. And he highlighted the climatological distribution of ocean acidification parameters in the North American coastal regions from Liqing Jiang. This is another work from Liqing that I wanted to highlight that came out last year that looked at the evolution of ocean acidification indicators in the global surface ocean. And so they actually combined the observational record with 14 different Earth system models to characterize the evolution of these indicators through the historical period and out to the end of the century. And these authors found overall agreement among the different Earth system models in projecting surface ocean carbonate chemistry once they were adjusted to the observational baseline. So it's a really interesting example of how observations and models can be combined to produce this interesting result. And the authors plan to update this work using upcoming CMIP7 model output and also expand this work to not just the surface, but the subsurface ocean. So other mapped data synthesis products that should be highlighted include those used in the global carbon budget. So this figure from the global carbon budget shows estimates of the ocean CO2 sink, estimated both through these surface CO2 fugacity products in light blue and global ocean biogeochemical models in dark blue. And there's a bit of a growing divergence between these two estimates that's driving a lot of research. But there are a number of different products of mapped observations that go into this product-based estimate. I'm just highlighting one here. This is ocean soda ETHC, which is a global surface ocean carbonate chemistry product from Greger and Gruber, which has recently been analyzed to look at global trends in ocean acidification indicators. Okay, zooming back in from the global ocean to a more regional scale, the work that I'm discussing in more detail today focuses specifically on large marine ecosystems of the United States, which are coastal regions that are characterized by similar biogeography and similar physical processes. So in looking at coastal acidification, I wanted to mention that coastal acidification represents a combination of the oceanic CO2 uptake that drives open ocean acidification, along with other unique coastal phenomena such as freshwater input, organic matter, respiration, coastal upwelling, other signals that cause variability on different timescales in these OA indicators. And our monitoring efforts of coastal acidification in US waters include large-scale coastal research cruises, like the East Coast OA cruise, West Coast OA cruise, that are repeated every few years, as well as fixed time series sites, such as the Cal Coffee time series, where observations are made at seasonal resolution over multiple years. Also, moored buoys that make continuous measurements in one location throughout the year contribute to our observing of ocean acidification in the coastal ocean, as well as surface underway carbon observations from volunteer observing ships and from scientific vessels. So the idea behind this work that I'll describe is that to gain insight into coastal ocean ecosystem status, as well as the spatial and temporal variability and the drivers of that variability and change, it's helpful to translate observation level data into synthesis products and useful metrics. So this work was driven by a collaboration with the NOAA Ecosystem Indicators Working Group, which is the group that maintains the National Marine Ecosystem Status Dashboard. So this is a resource that provides indicators of ecosystem health in different large marine ecosystems around the United States. And the indicators are displayed as time series, annual time series, along with some icons that show the long-term averages of the different indicators, as well as an analysis of recent trends. And the working group established these criteria for the indicators to include on this portal. And it was determined that ocean acidification kind of fits all of these different categories. But the other criteria that are necessary are these indicator data criteria. So the data underlying each of these indicators must be publicly available, regularly updated, long-term, adequate spatial and temporal coverage, and have a sufficient signal-to-noise ratio. So with regard to ocean acidification indicators, we were really interested in this adequate spatial and temporal coverage criterion, because the observations that we're using to develop these indicators tend to be scattered in space throughout each of these large marine ecosystems and irregular in time as well. So to do this, we're relying on the fact that seawater carbonate chemistry is both directly and indirectly influenced by different environmental properties, such as the ones listed here. So temperature has a direct effect. Chlorophyll can be a proxy for biological activity that has an indirect effect on carbonate chemistry, mixed layer depth, salinity, each of these environmental properties can kind of drive changes in sea surface carbonate chemistry. So this just gives an example of the direct effects of temperature, both on pH and PCO2. So for kind of the seasonal cycle shown in this point on the map, these are the changes in pH and PCO2 driven by those seasonal swings in temperature. And this strategy is the same that's employed for those global sea surface fugacity data products. But we're employing this on a regional scale to get a more regionally specific and regionally appropriate look at each of these ocean acidification indicators. So the kind of overview of the construction of these data products, which are called RFR LMEs, is that we're taking this observation level sea surface fugacity data. We're doing some machine learning, which I'll describe, to map indicators at a monthly resolution and a quarter degree spatial resolution. And then we're taking these regional averages of each of the indicators to get these regional time series that are eventually displayed on the National Marine Ecosystem Status Dashboard. So the first step in creating these indicator products is that we're actually spatially clustering each individual large marine ecosystem using environmental variability in parameters such as sea surface temperature and sea surface chlorophyll concentration. So these figures on the right show the variability in each of these parameters, and we're using Gaussian mixture modeling to cluster the region by that variability into these subregions that reduce the amount of variability that needs to eventually be explained by the regression models that we're training. And one nuance here is that Gaussian mixture modeling provides probabilities that each spatial data point belongs to a given cluster. So when we're eventually producing estimates of the sea surface CO2 fugacity, we can blend between these kind of hard boundaries of the clusters. So we're not getting hard boundaries in our predictions. So we're using SOCAT data along with these random forest regression models to reconstruct CO2 fugacity within US LMEs. And random forest regressions are basically ensembles of decision trees where we start with these input variables, and our goal is to get an estimate of CO2 fugacity. And when we get a large ensemble of these trees, we can use a subset for each of these inputs to produce a fugacity estimate for a given set of input parameters. So we train these regressions, we do some cross-validation to evaluate their performance, and then eventually we're using the proxy predictor maps with the trained regression models to map out sea surface CO2 fugacity. We're then comparing those fugacity maps. So this shows for the California current system, the kind of upscaling from the individual monthly observations into the mapped product. We're pairing this with total alkalinity estimates from empirical seawater property estimation routines. These are developed by Brendan Carter, and we're using these to calculate the remainder of the CO2 system for these ocean acidification indicators. And then I wanted to show where these show up on the National Marine Ecosystem Status Dashboard. So this dashboard provides a snapshot of progression of different indicators in US LMEs. So this shows the climatological oscillations. We have some different biological indicators. We have some human dimensions here. You can see the real drop-off in 2020 due to COVID. Physical chemical indicators is where the OA indicators show up. So we have sea surface temperature, sea level, and then if we scroll down a little bit, we get to these indicators of ocean acidification. So we're showing partial pressure of CO2, surface ocean pH, and aragonite saturation state on this dashboard. So in addition to those time series, the mapped indicators are also available via NCEI, and these cover 1998 to 2023 at quarter degree resolution in each of these individual LMEs, and at monthly temporal resolution as well. One kind of high-level result is that we are seeing pH declines in each of the 11 large marine ecosystems that we are mapping. And this is, of course, not unexpected, given the continuing trend of ocean acidification at a global scale. And generally, these trends line up fairly well with the global pH decline signal, although there is some regional variability in terms of the actual magnitude of each of the trends. So for this product, we did do quite a bit of validation and evaluation. And as one part, we compared moored buoy observations of sea surface PCO2 to the corresponding grid cell in the RFR LME data product, along with corresponding grid cells in more global scale mapped CO2 fugacity products. So this figure shows those results for the CCE1 time series, and this table shows the average difference between the buoy PCO2 and the corresponding data product PCO2. And our RFR LME is doing pretty well in terms of these differences between the observations and the modeled values. And even this version of the product that is trained without the buoy data itself is performing quite well. So I think this is indicating that if we're producing these types of map products specifically at a regional scale, we can better capture the local scale variability than products that are produced at a global scale. And the same thing can be seen for this Gulf of Maine buoy off the Northeast US coast. And when looking at all of the moorings that correspond or that overlap with the RFR LME data product, we're seeing in terms of the average difference between the buoy PCO2 and the corresponding data product PCO2, we're most similar to the mooring measurements compared to each of these global data products. So just highlighting a couple future directions for RFR LMEs. So we're looking to produce these in near real time dynamically in the future. So instead of just having the previous year of indicators available, you can actually get these in close to real time. So we're working hard to try to get that implemented. We're exploring using some higher resolution predictor data sets to produce these types of products in greater resolution than the quarter degree monthly resolution that they are currently available at. And we're working with a PhD student to conduct some model-based experiments to more accurately constrain uncertainty in undersampled areas as well. So just my last slide here, some key takeaways is that these regionally specific mapped indicators are more representative of local scale variability than global mapped products. The maps can provide regional context for ocean acidification studies, and the time series that are available on the NAMES dashboard provide a good snapshot of OA status in US coastal ecosystems. That's all I have. Thank you very much for listening.
Excellent talk, Jonathan. Thank you so much for your participation. And I'm going to ask Anna to introduce our next speaker, please.
Yes, thank you. I think we're saving all questions till the end. So I guess without further ado, our next speaker is Dr. Lauric Feugere, who is a postdoctoral fellow at the University of Quebec, working with Dr. Piero Colosi. His current projects aim at integrating the multi-omic response of several aquatic species to combine global change drivers to identify molecular pathways of interest to guide conservation and fisheries strategies of vulnerable marine species. So, Lauric, the floor is yours.
Hello, everyone. Thank you very much for the introduction. So today I wanted to talk about a project I've been working on at the University of Quebec in Rimouski, which aims at using comparative lipidomics as a tool to explore the sensitivity of different populations to ocean acidification, but also when it is combined with warming, and how we can use these tools to refine the management of fisheries and conservation. I'd just like to mention that this is a collaboration with the University of Quebec in Rimouski, Canada, and also the Department of Fisheries in Canada and the University of Aveiro, Portugal. So as it has been mentioned before, we have an issue of ongoing acidification, which will also worsen by the end of the century. Particularly, there's been already a drop in pH of around 0.1 unit since the beginning of this phenomenon, and it is predicted by the end of the century to drop by another 0.4 units in the worst case scenario. We know that ocean acidification has lots of mostly negative effects on marine animals, such as reducing their growth or eroding the shells, reducing the survival. It will alter the biodiversity, and all of these may have effects, for instance, for the economy. However, acidification doesn't occur on its own. As it has been mentioned before, for instance, you will have other stressors, and these will accumulate across many regions of the globe. So this is a map showing the cumulative impacts of anthropic pressure, including, for instance, ocean acidification, ocean warming, and you can also have overfishing or pollution. And two stressors of interest are the ocean warming and ocean acidification, because as you can see on this map, they will co-occur together. And one of the things is that you can have gradients and also hotspots of ocean acidification and warming, and this can shape the regional adaptation or, on the other hand, regional extension. So the question here is, what are the effects of ocean acidification when it is combined with ocean warming? We can expect that there will be some unanticipated effects because of interactions. So when you have two stressors and you add them together, maybe they will just have the sum of the individual effects. This is called an additive model. On the other hand, maybe you can have some biological surprises that emerge from interactions. For instance, you can have a stronger effect than expected, such as a synergy, or on the other hand, you can have a lower effect than expected, which is called an antagonism. This has been measured in different metrics at the whole organism level, such as behavior, survival, or metabolism, and it shows that there is a mix of synergy and antagonistic responses. However, these responses at the whole body level, they are underlined by cellular mechanisms, such as changes in metabolites, proteins, lipids, and genes, and we can use these as early indicators of the risk before we observe them at the whole organism level. One of those molecules are the lipids, which are key cellular actors because they have a polar head and a non-polar tail. And the lipids, we call them the lipidome when it is the sum of all the lipids in an organism. However, this lipidome is understudied. Nevertheless, it has key functions such as transport, signaling, and fluidity of the membrane, but they are quite sensitive to changes in the environment. And they will control the integrity, or on the other hand, the alteration of the cellular and subcellular membranes. And as a consequence, there could be changes of growth, immunity, or metabolism. And we can use the new tools of omics which give opportunity to find new findings because they measure a large set of molecules. And with this technique, we can understand some complex mechanisms. So we can go from measuring the genome to measuring the proteins, or here we can use a metabolite, including the lipids, to have a real-time view of the changes in the cells. That's why the aims of the study were to explore the potential of comparative lipidomics as a tool to identify functional changes in the lipidome, particularly in response to combined ocean acidification and ocean warming. And this would help us to identify whether there would be species or populations that are most vulnerable to these combined treatments. And then we can use this information for the management of fisheries and conservation of species. A good model to study this is the northern shrimp, Pandalis borealis, because it's a keystone species that is a prey for many other species. It also provides high-quality seafood, including for the lipids. However, the stocks are plummeting in North America. In North America, we have the Northwest Atlantic system, where the shrimp are facing different scenarios of ocean warming and ocean acidification. This means that there are different local habitats. Particularly in the Gulf of Central, the ocean acidification is quite strong, and it is combined with ocean warming. And this is due to changes in the currents. So we have here a good opportunity with a good case study using a previous experiment where they have looked at the combined effect of ocean acidification and ocean warming in the northern shrimp. And using integrative comparative physiology, using survival, showing it was reducing, for instance, to changes in the metabolome. However, we don't know much about the lipids, and this is the key point of my study here. So using comparative lipidomics, we want to compare different shrimp populations from distinct environments. We expected that there would be a stronger response when you combine ocean acidification and ocean warming. And we were hoping to use this information of population-specific responses as a tool to prioritize the conservation of the most vulnerable populations. To do this, we had the experiment design where we were collecting shrimp from different origins in the Northwest Atlantic. So we have the Central Estuary, the Eskimo Channel, Newfoundland, and Eastern Scotian Shelf. Then these were brought into the lab and exposed for 30 days to a factorial experiment of ocean warming and ocean acidification. So we had the conditions or favorable conditions of 2 degrees and no acidification. And then we looked at the effect of moderate ocean warming and a more severe ocean warming combined or not with ocean acidification. And you can note that there would be, because of this factorial design, 2 conditions of combined ocean acidification with moderate and severe warming. Then we quantified the lipidome from the abdomen muscle of the female shrimp. We had a high number of individuals. We used 270 lipids, and we looked at the changes of the lipid composition and particularly the function of those changes. I will just guide you through how we analyze the data so you can understand it more easily. So what we did was to compare the shared and unique changes in the lipid composition in the moderate warming and in the severe warming and also in ocean acidification. Then we looked at the combination of ocean acidification with moderate warming and ocean acidification with a more severe warming scenario. Then we looked at the changes, and if there were more changes in the unique condition, it would be a sign of antagonism. On the other hand, if you've got more changes in the combined condition, it would be a sign of synergy. And then we looked at the different functions associated to those lipid changes, such as the curvature of the membrane, the fluidity, or the thickness, as a way to understand how the membrane would be altered or not. So using this method, we were able to find that the combined effect of ocean acidification and ocean warming, they varied between the different regions in the Northwest Atlantic. Particularly in the population from the Central Estuary, it had the strongest evidence that there was a combined effect of acidification and warming together. On the other hand, the 2 populations here from the north, they were showing some partial effects of the 2 combined treatments. On the other hand, the population more in the south showed no effect of acidification, but only an effect of temperature. Temperature was showing an effect in all of the populations. So it showed that there is an interaction between ocean acidification, ocean warming, and the origins of the populations. Then we looked at each population, what was happening. So first of all, in the shrimp from the centurons, we found that in the moderate warming scenario, combined with acidification, it was inducing much more changes. So it was a synergism. However, this synergism was associated with a maintenance of the integrity of the membrane, which would be more of a positive response. Then when we go to a more severe scenario, the thing changed. Particularly now we have an antagonistic response, meaning that ocean acidification prevented the compensation to severe warming. And that was more of a positive effect. So the shrimp were highly sensitive, but they can maintain their membrane integrity. Then in the population of the Eskimo channel, in the moderate warming scenario combined with acidification, we had a slight limitation of the thermal compensation, which would be more of a negative effect. But there was again a shift when we were going to the more severe scenario, where in this case, there was a strong alteration of the membrane integrity in the combined treatment. So these shrimp would be most vulnerable to the combined scenarios of warming and acidification. The third population was the Newfoundland population. There were little changes only in the moderate warming. However, when we looked at a specific class of lipids, the sphingomyelins, these are associated with the regulation of salinity and also the immune response. We found that in the combined condition, it had a much stronger effect of this class of lipids. So it's a synergistic compensation response. It would be a positive effect for these shrimps. On the other hand, when you go to a more severe scenario, now you can see that the combined condition of ocean acidification and ocean warming led to an antagonism. Particularly, this meant that ocean acidification prevented the effect of the thermal compensation under severe warming. So the shrimp from this population, they are sensitive to ocean acidification, particularly under the severe warming scenario. Then the last and fourth population, the shrimp from the eastern Scotian Shelf, they showed no effect of ocean acidification. So instead, we looked at the effect of moderate and severe warming. And as you can see here, there were most of the changes happening only in the more severe scenario of ocean warming. So only the scenario of severe ocean warming induces a thermal compensation, and that meant they were able to maintain the integrity of the membrane. So that would be more of a positive effect. So these shrimps from this location, they are not sensitive to ocean acidification. However, they are sensitive to ocean warming and the more severe scenario of ocean warming. So in summary, what we can see is that we can use comparative lipidomics because they're evident that ocean acidification causes complex responses in the lipidome, and that these responses are specific to each region in the Northwest Atlantic. Moreover, we can see that there were changes and shifts from synergism to antagonism, or vice versa, when it was combined with ocean warming and when the ocean warming was closer to the thermal limit of the shrimp. So when we look at all of these shifts and we put them in the context of the membrane integrity, we can propose a scale for concern for the conservation. Particularly the shrimp that were more in the south, they were only sensitive to ocean warming, so they may not be impacted yet by ocean acidification, so they may be of least priority. However, the shrimp from inside the Gulf of the Centurons, we can propose that they are more sensitive, and particularly the shrimp from the Eskimo Channel here, they were showing the most vulnerability to combined ocean warming and acidification. And particularly, we know in the wild that these cannot move away from this condition. So this scale would propose that we should prioritize the conservation on this population specifically. But overall, what I want to show from this presentation is that we can use comparative lipidomics because they show variation in the interregional responses at the molecular level for a marine species that is exposed to ocean acidification combined with ocean warming. Moreover, we can use the comparison of the different populations at the lipidome level to understand which population may be more vulnerable to the conditions in the wild, having the knowledge of the long-term acclimatization. And last, we can use comparative lipidomics because they are a promising tool that we can use to inform and guide the conservation management strategies and also for the fisheries. Thank you very much for your attention, and I'd like to thank my laboratory at the University of Quebec and Rimouski and the University of Alberta, and also the members of the panel list.
Thank you very much, Lauric. Excellent talks also. We want to move on. Next speaker from Mexico is Doctora Laura Liliana López Galindo. She's from the University of Baja California, and she works for the Instituto de Investigaciones Oceanológicas. Laura is a marine biotechnologist with an interest in also in OA. She's working in several areas of the aquaculture, but she's working in the OA and temperature effect, the genomic response for marine mollusks during their ontogenetic development. Some of her current works involve using physiology, genomics, and transcriptomic tools to study the response for several organisms, bivalves, gastropods, about the oceanification in temperature. The floor is yours, Laura.
Thank you, Martin, for the introduction. Let me share my screen. Can you see my screen?
Yes.
Yes. Off. Okay. Thank you, Martin. Good afternoon, everybody. I will talk about a research that we have been developing in collaboration with other researchers here in Mexico and with other institutions. And the first thing is, as we know, the ocean acidifications refers to the continuous decrease in the ocean pH due to the absorption of anthropogenic CO2. This CO2 has been generated since the Industrial Revolution and has accumulated in the atmosphere. However, there needs to be a balance between atmospheric CO2 and the ocean's CO2. This is where the ocean acts as CO2 sink, as shown in the left image. We have here a representation of the CO2 parts per million since 1990 to 2020, and we can see the increase to 340 to 390 parts per million of this CO2 in the ocean. So what does this means is that the acidification in the ocean is now about 30%. And this has certain implications in marine mollusks. And how can we explain this? Is a simple form. We have the CO2 atmospheric, and it's combined with the water and form carbonic acid. Then this carbonic acid combines with the carbonate in the water and form bicarbonate ions. So this makes that the disponibility of the carbonate for organisms that need this carbonate to form calcareous structures, such as corals and mollusks, has problems to form their shales. So in this sense, the mollusks are one of the most representative phylum, with nearly 43,000 species, of which gastropods and bivalves are the most prominent. Both groups require calcium carbonate for the shell formation, which consists of aragonite and calcite in varying proportions. And one of the primary issues that ocean acidification poses for this organism is related to the difficulty of biomineralizing calcium carbonate. This leads to several problems, including impacts on metabolism, acid-base status, reduce the reproduction, the survival, and cause an immunosuppression in the organism. So this problem difficult the develop of adaptations for these organisms to this ocean acidification. In this sense, in Mexico, this situation has become particularly important because we experience seasonal occurrences of corrosive waters along the northern Pacific coast. This region is important as is the habitat of several mollusk species, such as the abalones. We have at least 4 species of ecological and commercial importance as the red, the green, the black, and the yellow abalone is distributed along the northern Pacific coast. The red abalone is one of the is one of the most important species and significant, and has suffered a dramatic decline in its populations due to 2 main factors. The first is the overfishing, that because this organism represents the 45% of global aquaculture, and we can see this in this image in the right, that is the annual aquaculture leap weight in tons that are from 1970 to 2020, and there are a logarithmic increase. So this has a vulnerability in these organisms by this overfishing, but too for the acidification in the ocean. And this causes mass mortalities during the early life stages due to the climate change and this synergy in between the ocean acidification and the climate change. In this sense, we have complex morphological changes in the life stages of the abalone, and these organisms have a planktonic lifestyle, meaning they live in the water column, and this cycle consists of 9 main stages: the embryonic development, the hatching, the belliger maturation, the competent larvae, and the settlement. And the post-settlement, that is the adult organism that is suffer a metamorphosis, and he is bentonic, or it is bentonic. So each of these stages represents a bottleneck in the abalone's life history, and they also have a benthic lifestyle, meaning they are found on the ocean benthos. And during their planktonic life, they are facultative lessitotrophs. This is meaning that they don't need to feed in the first days of life. They have hydrosolic filtration, changes in acid-base homeostasis, and they need a benthic microflora as natural inducer for the settlement, and they realize active biomineralization to produce its shell. But, however, it has been observed that when an organism is under stress, stress is there is a loss of performance which affects biomineralization processes or rates and erosions the shell. And there's a change in the cellular acid-base homeostasis in these organisms. This is important because it has been determined that populations forming aragonite are more sensitive. And in the case of the abalones, the early life stages depends primarily on aragonite. This in turn affects the survival of adult populations, which rely on the success of the early life stages. In this sense, one key observation is that most mollusk acidification studies focus on acute stress, and while ocean acidification represents chronic stress due to prolonged exposure. For this reason, we decide to evaluate the effect of 3 pH levels at a control pH of 8.1, a medium pH of 7.8, and a low pH of 7.6 under both chronic and acute stress conditions during different early life stages of the reed abalone. To achieve this goal, we designed a recirculation system that we can see in this image. And we have in this system we have a tank that we use to mix the CO2 that is injected with a solenoid valve in this place. It includes 6 2.8 liters aquariums and a compensation tank at the bottom that distributes the water, and aquamedic controller with an electrode that let us to control the injection of CO2 in the system. And we have 3 experimental systems that were used for this experiment. When we have this system, we place fertilized eggs under chronic stress at 3 experimental pH that we can see here in this image. We have the fertilized eggs, and we put in the control pH, in the moderate pH, and the low pH under chronic stress. And we evaluate these organisms in different stages, since 24 hours to 108 hours post-fertilization. And in the moment of the settlement, we include organisms that comes from the control pH at 8.1 and make experiment of acute stress, and these organisms were put in 3 different pH, the control, the moderate, and the low. So we have the 2 experimental assays at the same time, and we evaluate the performance of these organisms in the different stages. So for this, we have we evaluate with bright field microscopy the different parameters of ontogenic development. We evaluate the percentage of hatching, the competence percentage, the settlement percentage, and the survival percentage by different techniques like scanning electron microscopy, polarized light microscopy, and bright field microscopy. We evaluate the morphology of this organism, and we took in account the formation of the shell, the calcification, and some abnormalities in the organisms. Then we evaluate with Illumina massive sequencing the genomics part, and we work with microRNA sequencing to detect different microRNAs that are important for the developmental processes and biomineralization. And we like to identify another biological processes involved in this response to ocean acidification. So our results showed that a significant decrease in hatching percentage was observed at the low pH, and we identified a high percentage of larvae with abnormalities in their cells. This could be associated with atrophy in the calcification gland that this organism has to produce the mineralization of the shell, and the genetic machinery that regulates biomineralization processes, as well as the reassignment or reduction in the energy allocation for compensate the development in this organism. In this figure, we have the specific growth rate, where a significant decrease was observed in larvae at 30 and 48 hours post fertilization. And this shows that there is important the first stages of the lifestyle of this organism, and at the stage of 60 hours, there's no significant differences. And we think that this is because this organism has this lecitotrophic behavior. So is a form to compensate the stress caused by the pH at the 60 hours. Then we have, by the birefringence technique, the calcification rates. And what we see here is that in the control, we have a total calcification of the shell. But when the pH decrease, this calcification decreased too. So we prove that the 7 or the low pH has an effect or a severe effect in the formation of the shell. And even we have an organism without this shell in the low pH. Then when we talk about the settlement under the chronic stress, we don't observe differences in the settlement, but when we evaluate the effect in the shell with the electron microscopy, we observe that the normal granular texture with alveolar pattern is that we see in the figure A and D. So this is the pattern that we need to see in this figure. But when these organisms are exposed to this acidification or this chronic effect, we observe signs of shell deformations here in the protoconch and microfractures in different zones of the shell. And the alveolar pattern is altered by these different pHs, since the medium and the low pH. Then when we observe the organism in the acute stress, they don't have problems with the shell formation. The proto shell is very well formed. There's no evidence of abnormal granular texture or microfractures, but we can see a pattern of erosion in the figure F. So maybe the time that we have this organism in acute stress is not the sufficient time or enough time to show this deterioration, but we don't know if with more time we have the other patterns that see when we have the chronic stress. When we compare the settlement, we observe that in the chronic stress, we don't have problems with the settlement, no. But when we have the acute stress, there's a significant effect, not only in the low pH, in medium pH too. We have these problems in the percentage of the settlement, and these show that the larvae of the long-term acidification stress show a deterioration of the shell, but these not affect their settlement. And in the larvae exposed to short-term, indeed the acute acidification stress, did not show the shell deterioration, although the settlement was significantly affected. And we think that this is by the genetic machinery that is not prepared to this acute stress. So in this sense, we work with these microRNAs that are molecules that play a crucial role in regulating the gene expression. So they don't code for proteins, but instead bind to specific mRNA sequences to inhibit their translation or induce degradation of this mRNA. And in this case, a lot of RNAs participate in the larval and embryonal development of different species of mollusks. And in this sense, in this figure, we can see the mRNAs or the microRNAs that we identify. And we can see that in the control, we have at the 24 hours, the microRNAs that are involved in the developmental stages of this organism. And in the 110 hours, we have just a little part of these microRNAs, because this is the moment in that the organism is prepared to make the metamorphosis. So to take this advantage and be a benthic organism. But when we have this organism in the low pH, we have problems with the expression of the microRNAs. Practically, we have the same number of microRNAs overexpressed in 24 hours and in 110 hours. So this shows that there are microRNAs that are focusing on response to the ocean acidification. And in this sense, we work with different programs to evaluate the different biological processes that are affected by these different pHs. And as we know in the left figure, we have all the biological processes that are important to the first stages of the development at 24 hours, and we can see that there are a response to oxidative stress, cytokinesis, blood vessel remodeling that are important for the formation of the larvae in this process. And when we have this organism at the 100 previous to the settlement, we have these biological processes that are important to respond and to maintain the integrity of these organisms and prepare it for the settlement. But when we have in the right figure the different microRNAs that are present in the different stages that we evaluate, we have these overexpressed microRNAs, and we have a lot of biological processes that are affected for the ocean acidification. Now we are working in discover the different microRNAs that we have here. We are working in evaluate the different rates of expression, and we are making our article of this, and we don't have more information about, but instead we have different processes that are affected for the ocean acidification, including genes that has to see with the reproduction of these organisms. And it's important because if we don't have these larvae, we don't have adults, no. And well, what are the challenge and future perspectives that we have? First, well, we have to evaluate the several juveniles, and if these juveniles are capable of continuing their development until respiratory pores are formed, and whether they experience any growth limitations. We have to assess the biological processes and metabolic pathways affected by acidification stress, as well as the impact of on biomineralization. We have to conduct field studies on acid upwelling patterns in the field, like because the organisms there are in that place, no? And we have to determine whether hybrid abalone poses any advantages against stressors such as acidification, and we have to examine the phenotypic plasticity of green abalone under acidification and its interaction with the temperature and their synergy maybe. And well, we have a lot of collaborators here in Mexico. We have collaborators in CICESE, la doctora Fabiola Lafarga, which is an excellent person that works with abalone and is an expert in abalone. La doctora Claudia Ventura, Clara Galindo, here in the Ayon, Dr. Martin, he is such a great collaborator, Dr. Orion Norzagaray, Dr. Ernesto Larios, and we have a lot of students working on in these projects right now, and different laboratories that are working in elucidate what happened with this acidification and how these organisms has or not the mechanism to respond and to maintain it alive. So this is all for my part. So thank you so much for the invitation.
Thank you so much, Laura. Excellent talk. Also, we are so happy for all the presentations today, for all the speakers. And the next step is now to open the floor for questions. You can either type it in the question box or raise your hands. Anna, could you please follow with the question that we have in the box? Thank you.
Yes, we can also call on people directly if you raise your hand. But I guess I have a question for Laura. Have you done similar experiments under thermal stress yet, or is that just a future direction? And how do you expect thermal stress coupled to ocean acidification to affect kind of the results of your other experiments?
We have a project right now with ocean acidification in the green abalone, And we are working with transcriptomics right now. So the idea is to compare the microRNAs that we identified in the red abalone with the transcriptomics, and identify the objective of the focus genes to prove that instead the acidification has an effect on the different stages of the abalone life cycle. So we are working with transcriptomics too.
Excellent. Dr. Feugere, you mentioned that you have a question for John. Please do it.
Yep. Excellent talk, John. I really appreciated your vision for this. My question for you is looking into the future a little bit. What do you envision to be a reasonable plan for providing these indicators on a regular basis? Can you envision continuing the monthly estimates of these indicators going into the future?
Thanks, Dick. Yeah, I think sort of a monthly update would be a reasonable expectation for the future. The limiting step there is primarily, well, the SOCAT database is released on an annual basis, but we could certainly have regression models trained based on the previous year that we can apply to incoming proxy predictor variables. But the other limiting step there would be the availability or the update frequency of those proxy predictors. So that's driving some of our work right now in looking at the available data sets that might be beneficial to use going forward.
Excellent. Thank you very much. We have a question for Lauric. Lauric, you don't mention too much about the different species that you are using in your area for the shrimp. Some of them used to be more resilient. In Mexico, we have the species we call white, is Banamey, right? Used to be resistant to salinity, resistant to temperature. And what about the species that you're using for that? Are you considering also the fact of the salinity?
That's a good question, thank you. So in this experiment, we only considered ocean warming and acidification for what I've shown at the lipidome level, but we also have hypoxia, but we don't have salinity. So I'm expecting that that would be another stressor that would impact them. That the list of all the cumulative stressors would impact these shrimp, yes, including salinity.
Yes, John, please.
Yeah, sure. I had another question for Lauric, so I was wondering if I could just ask that. So you mentioned some recommendations for kind of management and conservation of these different shrimp species. I was wondering if you are participating in or in contact with, kind of the local management folks in kind of implementing any of those strategies or any of those prioritizations.
Thank you. And at the moment, we're not yet in contact with implementing those directly. Just wanted to show how we can use them, these comparative omics, as a tool for conservation, but we're still a long way from implementing them. I'm hoping in the future we would be able to implement these type of data at the cellular level and whole organism level to inform on the conservation of species of interest or population of interest, but it's still a long way to go here.
Cool, thanks.
Thank you, Jan, for your participation. Jan, you have a question also for Lauric. Please go ahead.
Yes, thank you. Oops, I guess I can, no, I can't turn myself on. Anyways, Lauric and to all of the speakers, thank you for great talks. These were really interesting. Lauric, my question is for you, and I was really surprised that the different shrimp populations had such different responses. And I'm wondering, were you surprised? And also, what do you think is going on there? Is it because that they're so isolated? I usually think of shrimp as being somewhat, you know, swimming around a lot. Forgive my ignorance of shrimp. I'm a oceanographer. And so I was just wondering if you could comment on that. Thank you.
Yeah, thank you very much for the question. So these inhabit quite different environments already in the centurions. There is some collectivity, but it's been shown that there were, it was last year in a paper showing that there are different populations, particularly the one from the centurions or the Gulf of the centurions seem to be genetically different. And these are the ones that are being impacted the most by acidification and warming. So they may be one of the reasons why we see these differences between the different populations.
Thank you. So was it a surprise for you or not? Were you expecting the differences?
I would say yes, we could expect those differences because particularly at least the separation between the Gulf and outside of the Gulf, where the condition inside of the Gulf seems to be the strongest. So I'm not surprised that there is such a difference here. But then if we were to compare more species, maybe there would be species that have, more population. Maybe some population would show similar patterns. But I would expect, I wouldn't be surprised if there are some groups that show important differences.
Thank you so much. Back to you, Martine. Thank you.
Yes, thank you so much. Elise, please.
Yes, I had a question for John related to, so you mentioned there have been declines in observations recently, and I was wondering if that was really only related or mostly related to funding, or if there are other things going on.
I'm not quite sure, and I'm probably not the best person to ask that, but I think it's potentially funding related. I know there was a recent paper, and I'm sure Dick can comment more, but by the a group of folks, I think, involved with SOCAT, who pointed out that the decline in the observational coverage leads to greater variability among the different products that are produced. So it is having, I think, a tangible effect on how well we can constrain surface ocean carbonate chemistry. I don't know if Dick wanted to comment more on that or had a separate question.
I was just going to point out that a lot of our observing system is dependent on ships of opportunity, primarily card carriers and other ships that transport materials between one country and another. And those ship lines have changed dramatically in the last few years. And it takes a significant amount of time to recover and find a new ship when the ship that you have utilized is no longer going at the same track line. So a lot of that's what's been occurring more recently. And this is one of the reasons why we're trying to establish some new technologies like sail drone and other approaches.
Thank you.
Gotcha. Perfect. Thank you.
I have a question also for John. John, in your approach, you mentioned you use the Carter methodology for calculate alkalinity. Which is your experience when you have rivers?
My experience is that the uncertainty increases. I think Brendan's algorithms, they do produce uncertainty estimates that, if I'm not mistaken, scale fairly directly with salinity and depth. So the river input does certainly increase the uncertainty of those alkalinity projections from the ESPER algorithms. And we do, I should have mentioned, we do have uncertainty with the gridded data product with at each grid cell, where we produce an estimate of each indicator, we also are producing an estimate of uncertainty, and that's based partially on the estimated alkalinity uncertainty, and then on an uncertainty that we're estimating as part of the CO2 fugacity estimation process as well. So yeah, rivers uncertainty goes up.
Okay, thank you. Well, I'm so happy to be today, the last day of a week, to share with all these speakers. Congratulations. Thank you so much. Thank you, Dr. Feely. And the floor is yours for the final minutes and final message, Elise. Thank you.
Yes, thank you so much, Martin. A big thank you to all the speakers today. Such great talks. And also a big thank you to Anna and Martin for being our moderators. And we want to thank you, the audience, for joining us for OA Week 2024. Please don't forget to consult the website. There's only one more session left for OA Week 2024, which is coming up in about 30 minutes, so we would love for you to join. It's an iconic session talking about different career paths within the ocean acidification field. And also, if you would like to stay up to date with the GOA-ON community, consider signing up for GOA-ON in the link that Katerina put in the chat earlier. And then also, I just wanted to share that we have an open call to join the GOA-ON Executive Council. Go ahead, feel free to scan this QR code for more information, and feel free to nominate yourself or someone else. And thank you, everybody, so much for being here with us, and we'll see you next time.